Good morning to one and all. Thank you so much for joining us. And so today's speaker is Dr. Pradeep Kumar Singour. So I just want to give a brief introduction about Dr. Pradeep Kumar Singour, sir. So, Dr. Pradeep Kumar Singhal is a highly accomplished pharmaceutical scientist with an extensive academic and research career spanning over 20 years. Currently serving as a professor and vice principal at Venus Group of Institutions, Faculty of Pharmacy in Bhopal, Madhya Pradesh, India. Dr. Singhal is renowned for his expertise in pharmaceutical chemistry and drug design. He completed his B Pharm and M Pharm in pharmaceutical chemistry from Dr. H.S. Gaur, Central University, Sagar, and later earned his PhD in pharmaceutical sciences from the same institution. Throughout his career, Dr. Singour has made significant contributions to the field of pharmaceutical research with a particular focus on rational drug design. His research interests encompass various areas including virtual screening, de novo design, QSAR, analysis, molecular docking, conformational analysis, biochemistry, and medicinal chemistry. He has authored over 43 research and review articles published in prestigious national and international journals, demonstrating his commitment to advancing pharmaceutical science through cutting edge research. Dr. Singh's dedication to teaching and mentoring is evident through his supervision of more than 40 MPharm students, the guidance of two PhD students in their research projects. He has recognized for his outstanding contribution to education and research, receiving awards such as the Various Employee of the Year Award in 2019, the iconic Faculty Award from Shiksha Sansthan Jaipur in 2019, and the Excellent Teacher Award in Pharmacy by Sister Nivedita Takniki Shiksha Samiti Bhopal in both 2013 as well as in 2018. Additionally, Dr. Singour has shared his expertise by delivering expert talks at various conferences, serves as reviewer for reputed international journals, further establishing his prominence in the field of pharmaceutical sciences. We all welcome you, sir, for the today's webinar on computational approaches streamlining drug discovery. So, welcome you, sir, and thank you so much for joining and giving your precious time and sharing your time with us. Thank you so much, sir.
Uh, Thank you, Mr. Monu, and thank you for introducing me among the participants. Uh, Today, we will discuss about the different computational approaches that will streamline the drug discovery, because we know what are the drug discovery, and there are different steps involved in drug discovery, where the timeline of the discovery is high. So, by the help of different computational approaches, can we cut the time, and also we can reduce the time, and as well as reduce the cost. So here we will discuss only some brief of that computational approach, because there is a wide areas of the data discovery and the computational approaches. But here we will only that uh, discuss some approaches. What are the approaches, and I will give some uh, how we can perform the computational approaches. So we will discuss, uh, as we know that the life expectancy is now increasing, because there are different drugs have been discovered, and also there are the new drugs have been discovering. So we have to involve in different approaches of the computational approaches, because the era is the era of the computational technology. So we can combine the computational as well as the technology, so we can discover more new drugs, so we can all increase the life expectancy also. So, and we know, I think all we know about the what are the adventures in drug discovery. For the development of the one drug, we have to start from the 10,000 to 30,000 compounds, and we have to synthesize, examine, and then screen, and then only 10 to 20 compounds come into the preclinical tests, and then we have performed the different clinical trials on phase 1, phase 2, phase 3. and finally we got only one compound. So the time required for the discovery also approximately 15 years, and the cost of the compounds will for the production, for the development, will also high, because we have to start from the 10,000 30,000 compounds, and only we get only one successful compounds. So how we can cut the time and how we can reduce the cost by help of that computational approach, we will discuss today. um, I already discussed that any drug come into the market after starting from thoughts and starting from 10 to 12-year back, 15-year backs, and we reached only the one compound, that approved compound. So, what are the different challenges of the medicinal chemist? Because that if you want to discover any one compound, that they have to increase the costs and enormous time. That we can, by the help of computational approaches, we can, how we can develop the successful compound by reducing the time. So these the drug discovery and development time, how we can discover that. First we have to identify the disease, and then these are the traditional approaches by which, first we have to identify the disease, and then we have to isolate the proteins that involve in particular disease that require 2 to 5 years, and then we have to find the drug that is effective in that particular disease, and particular proteins and particular receptors that require 2 to 5 years, and then we have to perform the preclinical testing that's also required some years, and then we have to perform the scale up, then file IND, and then we have to perform the human clinical trials, and then file NDA, and then require the FDA approval of the drug, that require 2 to 3 years. So we can see here there are the lots of years are required to develop any drug discovery. So how we can reduce some years by the help of that some molecular modeling techniques and the virtual screening, that's in to find the different drugs that is effective in the particular disease. So what are the different molecular modeling techniques, and what are the different virtual screening techniques? Molecular modeling, by name itself, that's modeling of the molecule. We know that modeling, we it is a general words, but more, if you perform the modeling of the molecules, it means the molecular modeling. And by the help of virtual, by the help of computer, if we screen the lots of compound, known as a virtual screening. So by the help of virtual screening and the molecular modeling techniques, we can design the drug, and we can get the effective drugs in very lesser time. So what are the different computational approaches? Because we are discussing today about the computational approaches that streamline the drug discovery, means how we can drug discover by the help of that computational approaches. So where we can involve, so we can see that if you want to isolate the proteins, so by the help of genomics, proteomics, biopharmaceuticals, we can um, isolate the proteins by the help of that study of the genes, that genomics, study of the protein, that the proteomics. So by the help of these methods, and also by to find the drug, so we can screen the 1 lakh compounds at a time. That is high throughput screening. That lot of compound we can screen which compound will be effective and which compound is not effective. So we screen by the help of high throughput screening, and virtual screening by the help of computer. Virtually we can screen the lot of compounds, and how we can reduce the time of that synthesis by the help of computational chemistry. So computational chemistry, computational chemistry. So computational chemistry, it means that if we synthesize the compound in a combination, so we can reduce the steps. Suppose we want to synthesize the peptides. So peptides require that combination of the different amino acids. So can we reduce the time of that coupling of the reactions, of the removal of the coupling agent, removal of the protecting agents? So we can combine these steps in a one steps, so we can reduce the time. So that is known as a combinatorial chemistry. It means that chemistry we can perform in a combination. So we can reduce the time. It will help to reduce the cost of the development of the drug. And that molecular modeling, we already discussed that modeling of the molecules by the help of computers. So we can develop drugs. And also, there are different models in silico model of the ADME. We know the ADME, that's absorption, distribution, metabolism, and excretion. So there are different model like CCADME model, and then also there are some toxicity models are also available in which we can determine their absorption, pKa value, also that metabolism, also that their toxicity profile. So all we can there are different softwares are available, so we can screen out the compound which will absorb in the body or not. So now we come into the different drug discovery process. First, we have to we that discussed earlier that first we have to identify the targets, and then after identification we have to validate the target. Then we can perform the high throughput screening. Then after throughput screening, we can um, discover the lead compound, and then after evaluation of the lead compound, and then we validate the lead compound, and then finally we can come into the clinical trials. So after completion of the clinical trials, we can get the most effective compounds. So these are the process. And also the high throughput screening can be covered by the chemical library. We can use the chemical library that by the natural products. So natural product chemical library of the compounds that can be help into the high throughput screening. And drug discovery process, we discussed that by the chemical library help of the chemical library, we can use the rational designing, and then molecular modeling approaches, and then virtual screening, and the early ADME and toxicity profiling. So these are the different computational approaches by which we can develop a new drug. So where we can use and which type of techniques. So suppose there is a therapeutic target is there. The therapeutic target, how we can identify the therapeutic targets. So by help of that functional genomics, and then structural genomics, and then proteomics. And for the functional genomics and structural genomics and the proteomics, there are different softwares are also available by which we can develop therapeutic targets. And suppose you want to develop that, you identify the targets, and then you find the novel ligands, and how we can find the ligands, then by the help of structure based drug designing, and then chemoinformatics and high throughput screening. And and then suppose you come into the clinical candidates. So clinical candidates, you have to perform the in vivo disease model. There are different type of models are available. And then admit and drug delivery. Admit means ADME, that is absorption, distribution, metabolism, excision, and T means toxicity. So these models are also available. And then we can come into the medicine, that find you the best medicine. So these are the different approaches, and each approaches are used for the different purpose. Someone is used for the uh, development or identification of the target, someone is used for the ligands identification, someone is models are used in the clinical trials. During the clinical trials are the clinical candidates, that's in vivo disease model, admit, preclinical. So there are different approaches. Here I am only giving some brief, actually, just these are the methods, because every methods have their details, and we cannot discuss here because and there are different steps are involved for the every approaches. So only we are here discussing about the approaches only. And molecular modeling approaches. So, molecular modeling means the modeling of the molecule, and by the help of different approaches, just like that NMR and X-ray determination. By the help of NMR, X-ray structural determination, we can identify the proteins, because first we have to isolate the proteins, and then we have to determine the structure of the proteins. So if you want to determine the structure of the protein, we can determine by the help of NMR techniques and X-ray determination. And then we have to suppose we have to perform the energy minimization of that particular proteins of the or the ligand. So we have to perform the QM/MM method. QM means quantum mechanics, and MM, that is a molecular mechanics method. So quantum mechanics and molecular mechanics method, these methods are used to generate the different uh, candidates or the ligand, that by the help of energy, we can say that we can reduce the energy, we can minimize the energy by the help of these methods. So we can get the low energy molecules. Why we require a low energy molecule? Because it is a stable molecule. Because when drug is interact to the receptors, it should be in a low energy state. So how we can perform the energy minimization by using this energy minimization calculation, energy calculation method, that is quantum mechanics and molecular mechanics. And there is some homology modeling. Right side, we can see that homology modeling. Suppose you don't know the actual structure, how we cannot determine the structure of the protein by the help of NMR X-ray, then we can perform the homology modeling. If you know the structure of the receptors of the same family, so we can design the homologous structure of that same family. So we can draw manually the structure of the receptor by the help of homology modeling. Then we can perform the further study. And here we can say in upside that QSR and 3D QSR, that is ligand based drug design process. That QSR, that is quantitative structure activity relationship. If we have the large number of compound in the same series, so we can perform the SAR, but quantitative SAR. So we can get one equation, and by the help of these equation, we can determine the biological activity of the unknown compound. So later we will discuss in detail. And there is in low side, that is combinatorial chemistry. So already we discussed, if we perform the some reactions in a combination, so we can reduce the steps, that is known as combinatorial chemistry. And that is another one is bioinformatics and informatics, in which that is large number of molecules we can get their data by the help of bioinformatics. So what is molecular modeling? Molecular modeling, that is a visual representation of the molecule and their property, and by the help of these property, we can perform the drug designing process. And computational approaches and chemistry, that is simulation of that molecular property of the compound through the different equations. These are the equations, and we have to perform the statistical modeling also, and then there are different informational management of the database of that large number of the property. So these are approaches also used in when we perform the molecular modeling. Okay, so which approaches will use when? So we can see this table, in which so when we see that drug receptor interactions, there is some receptor is there, and some is drug or a ligand is there, and the drug or ligand is bind to the receptors. So suppose there is you don't know the structure of that receptor, and you don't know the structure of the ligand, then which method you follow? So we can see in the first column is there. So we have to perform that high throughput screening and the combinatorial chemistry, in which we can synthesize a novel compound in earliest step, and we can screen the large number of compound, which compound will act to the will receptors. So this method is used when you don't know the structure of the receptor as well as the structure of the ligand. And right side, you can see that if you know the structure of the receptor, but you don't know the structure of that ligand, then you can perform the de novo drug designing. Later, we will discuss in detail de novo drug designing, in which that if you know the structure of the receptor and we uh, build the structure of the ligand inside the receptor, that is known as de novo drug designing. So later we will discuss in detail. In this, we can see that suppose you don't know the structure of the receptor but know the ligand structure, so we can perform the ligand based designing. Ligand based designing, that QSR approach, pharmacophore based approach. So QSR, also 2D QSR, 3D QSR. So these approaches can be used. These approaches develop by equation, and by the help of equation we can later, by the help of equation we can uh, estimate or determine the biological activity of the unknown compound. And here the last one, if you know the structure of that receptor as well as the structure of the ligand, so we can perform the molecular docking. Docking means you can, in general word, fitting. That there is one structure of the receptor and there is one drug, and drug will fit to the receptor. So by which we can see that what are the different bonds are involved to the fitting, and how much that fitting is there. It is good fit or not fit. So that is we can see from the molecular docking. So we can screen the compounds by the help of molecular docking techniques also. Here also some more techniques which or we can see computational approaches that's used in a different purpose. Suppose that if you want to calculate the physiochemical property of that compound, so we can, by the help of there is some software and available, its apps are also available, in which you can calculate the log p, dissociation constant. And for the drug designing, there are ligand based drug designing. The ligand based drug designing is performed by the help of QSAR, pharmacophore based drug designing. And structure based drug designing, that is by the help of docking, de novo drug designing. And there is some pharmacokinetic modeling also, by which we can determine the absorption, metabolism, distribution, toxicity profiling of the drug. And chemoinformatics, by chemoinformatics, that is database management, similarity search, different methods are available for the chemoinformatics. So all other, if you join all the methods, so we can perform the virtual screening protocols, so we can develop new drugs. Here I am giving some name of the softwares that is used for drawing tools by the CameDraw, Marvin Sketch, CameSketch. And these some softwares are freely available in the internet, so you can just download and then we can use by the help of CameDraw, Marvin Sketch, CameSketch. So these CameSketch, these so we can draw the structure, because if you want to molecular modeling technique, if you want to perform the QSR approach, you want to perform the docking approach, then first we have to draw the structure. So by the help of these software, we can draw the structures. So you can just write down all the name of the softwares by the help of which we can draw the structure of the compounds. These are the some molecular modeling techniques or software, that is charm, that is bromex, amber, cis parameter, then cis side chain. So there are different techniques or software, you can say, that by the help of these we can perform the molecular modeling. For the binding site prediction, uh, suppose you know the structure of that receptor, and you find out the you want to find out the binding sites. So by the help of these different softwares, you can predict the binding site. So you can just write down docking. So I'm just discuss what is docking. So for the performing of the docking, we can use Autodock software, Dock, Gold software is there, and docking server. Schrodinger software is also available in which we can perform the docking study. So kind of target prediction, and these are the softwares. ADME toxicity, we can perform by the Volsurf, GastroPlus, MatCam Studio, LOPS, CCADME. CCADME is freely available. You just write down the CCADME. Just you can type the CCADME in the Google, and just you can see the one website is there, and you put the structure, and then you uh, these are the different softwares. Now come into that QSR. Here I am discussing few techniques in detail. So QSR, that is quantitative structure relationship, in which we have to develop a mathematical relation. We have to develop an equation between the biological activity and the physiochemical properties. So we have to generate the equation by the help of physiochemical property. There are different physiochemical property like the lipophilic property, steric property, hydrophobic property, electronic property. So these first we have to calculate the these property of the compound, and then we have to develop the equation in between the biological activity and the physiochemical properties. So there are different type of QSR, that is classical Hens analysis, 2D QSR, 3D QSR, and 3D QSR molecular field analysis, molecular shape analysis. So there are different techniques are available. So in QSR, suppose you want to perform the QSR study. So these are the steps of the QSR study. First, you have to select a training set. First, we have to identify a series in which there is a more than 20-25 compounds should be there, and we have to divide the data into two set, test set and training set. And first, so suppose you select the training set, then you enter the biological data in the software, and then we have to perform the energy minimization property, energy minimization, that is generate a confirmation, that is energy minimization, energy minimize conformer, and then we have to calculate the descriptor of all the energy minimized molecules, and then by the selection of statistical method, we have to generate QSR equation, and then after generation of the QSR equation, then we have to validate the QSR equation by the help of test set. We can generate the model or equation by the help of training set, and then we can validate the QSR equation by the help of test set. And then finally, uh, if you develop the QSR equation and validate the QSR equation, then you can predict the activity of that unknown compound. Unknown compound, we can predict the help of that QSR equation. So here I'm just showing how we can draw the structure, and so we can draw the structure, we can draw the benzene and then bonds, and we can draw the structure. Then finally we can come into that came 3D software, and here we can minimize the energy of that molecule, and then we can find after then we calculate the properties. Here is some list of that properties, and then alpha polarizability, band energy, boiling point. There are different properties, so we can calculate, and after calculation we can develop the QSR equation. So what are the different properties? What are the different descriptors there? So there are different descriptor like structure descriptor, electronic descriptor, thermodynamic descriptor. So there are different descriptors are available. Descriptors means properties. So we can calculate the different properties by the help of particular software, and then we can develop this type of QSR equation. So in which log 1 by C, it means that biological activity, and we can develop a QSR equation and biological activity and physiochemical property. So by the help of those, so we can see here some physical property will have the positive correlation, some will have the negative correlation. So these are the this is the QSR equation. So in which we can uh, calculate the biological activity means log 1 by C of that unknown compounds. If you know the pi value and the sigma value of that particular unknown compound, then we can put the value in pi and sigma, and then we can calculate the biological activity. Molecular, that is field analysis, shape analysis, that is molecular field analysis, in which that we have to align all the molecule, energy minimize molecule, and then we have to put the all molecule in the grid box, and then we can calculate the field, and that is known as a comparative molecular field analysis. In short, that is COMFA analysis, that is a CDQSR approach, in which we can calculate which area of that molecule is prominent. Suppose that is red color is there, so we can see here the red is indicated electronegativity. It means there is a, if you increase the electronegativity at particular area, so we can develop a potent compound that have the maximum activity. In blue color indicated that is lactopositive, it means you can put the you can add the group that have nature of the lactopositive. So if you add in this area, so we can develop a novel compound. And just like we calculate the steric field also, in which green color indicated someone, and that yellow color is, it is the repulsive steric field. So by the help of this color, that is known as a contour map, in which there is contour color is indicated. Yeah, we can say there's contour maps by known as contour maps. So by the help of that contour maps, we can uh, add particular group at particular area, so we can develop a best rep. So first we have to develop these as a contour maps by that help of that comparative molecular field analysis. And some more we can see here the some more properties there, steric property and the electrostatic. First we have to align, and then we can calculate the field. And also that COMSIA analysis also now that is comparative molecular similarity index analysis, in which also use that properties, these properties also. So another technique, that is a pharmacophore approach. And pharmacophore approach, there are different type of pharmacophore approach. Pharmacophore, it means the active part of the molecule that is responsible for the pharmacological activity. So by the pharmacophore approach, we can identify the active parts, identify the pharmacophore parts, so we can develop a better drug by the help of these pharmacophore approaches. So these are the different steps of that pharmacophore process. We can by the training, first we have to training set selection. Just I have discussed in a series, we can divide the test set and training set. Then after training set selection, then feature selection, which features you want, and then conformation generation means energy minimization generation, and then alignment of the all the molecule. Alignment means you have 20 compounds in a training set, so you have minimized the energy of the all the 20 compounds, and then we have to align all the molecule over to each other. Then after alignment, and then we can develop a model, pharmacophore model, and then by the help of that pharmacophore model, we can develop a new drug. So this is known as a pharmacophore approach. And pharmacophore features, there are different uh, uh, HB receptors, donor, hydrophobicity, hydrophobia, aliphatic, aromatic. We can judge, check, we can calculate that positive, here is a positive charge, negative charge. So by the help of these features, we can develop a model, and we can calculate the develop new compounds. So these are the steps of the pharmacophore generation. First, we have to input the structure, then conformation modeling, and then we can, with the SAR data, we can generate the model, and finally, we can evaluate the hypothesis generated. Now, coming to the next technique, that is the docking techniques. Docking, it means fitting, we can say. So, first we have to download the proteins from, that is one website, that is RCSB, that is the protein data bank. Just you can type the protein data bank in Google, and you can download the structure of the protein, and then we can perform the docking study. Then when you download the protein, then we have to optimize the protein by removal of the water molecule, adding of the hydrogens, and then, so it means that is known as a preparation of the proteins. So first we have to perform the preparation of the proteins, and then we have to perform the preparation of the ligand, means drawing of the structure of the ligand, and then preparation of the ligands, and then we can perform the docking study. Docking study, it means that drug will interact to the particular binding sites of that proteins. You all know that protein is made from the amino acids, so that ligand will bind to the particular uh, binding sites, mean particular amino acids, and if you form the more hydrogen bond with that particular protein, it means drug is most active compound is there. So by the help of these docking study, we can determine which compound is docking well, or which compound is not docking well. So there is some docking score is generated in a docking study. So after that, just like that, you say this cricket score is there. Cricket score, more score is means more run is there. Just like that in docking is there. There is some docking score. So better docking score indicated that the compound is fitting good to the ligand, or fitting to the good fitting to the receptors. So by the help of that score, we can check which compound is most fitted and which compound is not fitted. Another technique, that is a de novo drug design technique. De novo drug design, suppose you know the structure of that protein or a receptor, but you don't know the compound, which do not know the structure of that compound. So we can build the structure of that ligand inside the protein by the help of de novo drug design. De novo design, suppose there is a structure is there. These are the, suppose this is a structure. So we can develop inside the structure of that ligand. First, we can add a small group inside the protein. If he is binding to the receptor, then we can add the more groups or a ring, and then finally we can get a final compound that is most fitted to the receptor. So it means that building up that ligand inside the pocket of that particular receptor is known as de novo drug design. So there are different softwares are also available. So these techniques are the computational approaches. By this, we can develop a new better compound. So and now, what is a virtual screening? So virtual screening, it means there is, suppose you have a large number of compound, and there is a large number of compound in a library. So if you want to screen out which compound is active and which compound is not active, So we can screen the compound virtually by the help of in silico models, or by the help of computer, you can say. So we can screen, that is known as virtual screening. So virtual screening, suppose you want to, there is some natural compounds is there, lots of compound is there, natural compound. So we can screen the compounds. Now, the virtual screening is also now helpful in the repurposing also, because you know the compounds already developed in the market, but it is used for the some other purpose. So we can screen that particular drugs for the another purpose also. So it may be act to the another receptor or a proteins also, and you can, the particular drug will can also be used for the another purpose. So by the help of that repurposing, we can reduce the time of that production, reduce the time of that synthesis of that particular compound, because that particular drug is already in the market, but we are developing that particular drug or drug for the another purpose, another disease. So these repurposing is also using this these days. So they are scientists are developing that, scientists are searching which already developed drug is using it for another purpose or not. So another technique, that is a pharmacokinetic modeling. Pharmacokinetic, we know well that ADME is there. So that property relationship, there is some structure property relationship or not. So here we can say that most of the drug in the market are withdrawn because there is some commercial reason, other case some shows the adverse effect, someone is shows that toxicity, but most of the drug is not in the market due to their pharmacokinetic property, or that means there is some problems in the pharmacokinetic property. Drug is most effective to the particular receptor or enzyme, but there is some lacking to the property of that pharmacokinetic. There is some problem in the pharmacokinetic property, which means ki, suppose drug is not absorbing inside the body, so it is not in the market. But if you check that particular drug is binding to the receptor or not, it is better drug, but it is not in the market due to their pharmacokinetic. So we can check that pharmacokinetic modeling technique, that through the molecular modeling technique, we can check the pharmacokinetic property. So we can work on that particular area. Either we can enhance their pharmacokinetic property by any way. So the drug can come into the market. So uh, you can see that, suppose there is compound is good. So you can see that is good ligand because activity is optimized. We have performed the docking study and we find that particular drug is most active, good active. So this is known as a good ligand. But when the good ligand is converted into the good drug, when the activity as well as the property both are optimized. It means that physiochemical property, pharmacokinetic property. So when we can say the drug is good, when the drug is binding to the receptor as well as it have a good pharmacokinetic property. It is good absorb from that inside the body. It is not eliminated the body, and it is that is bioavailability is good. So these are the properties also matter when we develop the drug. So we can, by the help of the different pharmacokinetic model, we can develop a good drug. So what are the different property profiling assay we have to perform? That is, we have to perform the solubility study, permeability, bioassay, blood barrier crossing or not, cytotoxicity, transporter, metabolite study. So these study we have profiling of the drug, because drugs should must be survive, that there are different in vivo barrier inside the body. So because you know that in a stomach there is pH is 2, so its drugs should be stable in stomach. Drugs should stable in the intestine. It should cross to the intestine, and it should circulate in the blood, and blood have the proteins, so it should we have to check that protein binding of that particular drug, and there are renal clearance is there, how much drug, how much time the drug available in the blood. So we have to check by the help of different techniques and the models in for the development of the better drug. So each state of the biology has a property issue, just discuss, because suppose you perform the high throughput screening to purity, solubility, identity. These are the issue when you are performing the enzymatic assay, that stability in the bioassay and solubility. And then as you perform the cellular assay, then permeability, solubility, stability. And suppose when you perform the animal study, and then the drug is you want to give to the human, to then we have to check that that plasma stability, J stability, solubility, permeability. So these are the different uh, method or a technique that use that particular assay, and then we have to check if particular drug is stable or not. So there are different models and approaches all can help to check these properties. So there are some successful drugs that came into the market after drug designing process by the help of different computational approaches. These are the drugs that discover for the different purpose. Just like that, erythromycin analogs developed for the antibacterial activity by the help of that computational approaches. So these are the list. Some list, there is more drugs are also available that develop through that computational approaches. These are the some list. So, so finally, what is the drug discovery? Drug discovery is a multidisciplinary and that is complex and costly, and some that is intellectually expensive process. But modern drug design techniques can make drug discovery process and then fruitful and rational. Means that is computational approaches. By the help of computational approaches, we can discover the more fruitful and more effective drugs. and and the knowledge of these techniques, expertise in these techniques, we can save the time as well as the save the cost of the development of the drug, because we can reduce the time, we can reduce the process of the discovery by the help of computational approaches, so we can reduce the cost of the development of the drug. Computational approaches, or we can join the different computational approaches for the development of the new drug. So finally, some softwares are also here. Just I have written these that is Tripos Compa are used for the compa analysis. Volsurf catalysts are also used. Serious and that QSR Plus is there. Schrodinger is there. Discover software is there. uh, Wheel Life Science software is there. So there are different softwares are available, and the cost of the software it is dependent on the feature of that particular software. So some is used for the only 2D QSR, somebody is used for 2D plus 3D, and some software, by the help of some software, we can calculate, we can detect the compound of that. We can check the last of there is so many techniques. So by the techniques or the by the features of that particular software, there is some difference in their cost. Some software are freely available in the internet, so we can start from that part. software available, and then we can, if we have the expertise, then we can get that uh, these type of software. We can purchase these type of software. So thank you very much for listening me. If you have any query, you can ask.
Thank you so much, sir, and for sharing your experience with us. And so I want to request all the participants, if you have any query, just let me know in the chat box, or you can unmute yourself and you can ask directly. I think, sir.
Hello, sir.
Mm, Haji.
Uh, sir, when we are talking about molecular docking, you told us that it will generate a docking score.
Yes,
So I have used Autodock. It will generate like 8 models and different docking scores.
yes,
But those protein when I docked in Pymol, these proteins are very much far apart, not touching together. So how would I like
okay. It means there
understand
is
it?
yeah, difference in the results. You can say you are saying about the
Yes, sir.
difference in the result and Pymol and the dog or to dog, just like
Yes,
that.
yes,
Yes.
yes.
So we have to validate, and we have to optimize if these type of results are obtaining. Some software is saying different thing and some software is different thing, so we have to optimize by the help of different or more effective softwares. That we can use the glide software from the Schrodinger. So by the help of these, uh, we can check which compound is fitting or not, because definitely there is some variation in the results obtained. So we have to optimize and we have to validate from by the help of different more approaches or the different more effective software, we can say. So this is the we have to solve, we have to validate.
Okay, so and I have another question.
Yes,
Sir, I was trying to dock 2 proteins. So
yes.
one I selected the larger protein as macromolecule and the smaller protein as ligand. So while docking this the after docking, the autodock will prepare a protein, but it will have only small number of amino acid, not the whole protein. So how to check whether matlab, if my thing is कि I want to see where are those two are binding. So can we do anything to show to see where these 2 proteins are binding actually, or have another binding sites?
Either we have to check. We can first we have to check from the different software. I think we can check by that. I'm not working in that particular binding of the two proteins, but I think we can check by the most effective. I think that you can use the glide software. That is better one. So you can perform these study, I think.
Okay, okay, sir. Thank you.
Okay, okay.
Sir, we have another question
Yes.
from someone, GS. Good morning, sir. How is the drug design market at present? Also, what are the opportunities for drug design careers as bioinformatic graduate?
Uh, now these days, different companies is developing some. We can say that molecular modeling or the computational area. Just you can see just like the formulation development, AMD, and just like that formulation AMD, they are in the R&D department. There is one other computational uh, development area is there, in which they are using these techniques to develop the new drugs. So some companies which is working, which are working in that area of that development of the new drugs. So they are emphasizing to use the molecular modeling technique. So definitely, if you work on that drug designing, so it will help and you to get that job in that company that work in that particular field. So some companies you have to search which companies are involved in that in these area or techniques, so you can get your career in that particular uh, area and in particular job in the company.
Thank you, sir. And another one is, it's not a question, but it's a query. Sir, can you give us a complete list of free softwares and licensed softwares available for molecular modeling? Okay, so we will provide the recordings to everyone. Okay, so sir already presented all the softwares and list of softwares and licensed softwares. So you can extract the data from there and okay. So and sir, there is no further question from the participants.
Okay.
Okay,
Thank
sir.
you for
Thank you so much, sir.
everyone for listening
So
me.
I would like to welcome Dr. Karthikeyan from IGNTU. He also joined us this webinar. So I would like to invite Dr. Karthikeyan for vote of thanks and share your experience with us about this webinar. Sir, am I audible? Yes, sir.
Yeah. Good morning, all. Thank you, Mr. Monu Shukla, for providing me this opportunity.
Thank you, sir.
I would like to express my profound gratitude to the speaker, Singour sir. Hopefully, I think we will meet sometime when I come to Bhopal. And
Yes.
it was an enlightening session on drug discovery. And he had explained it so in a simple and thought-provoking manner. I hope that all the participants will be benefited from his lecture. And I am looking forward to the next lecture. So with this, and also I would like to thank the participants for asking, for being so interactive. And thanks again, Mr. Molo.
Thank you
Thank
so
you,
much.
Karthikeyan sir. We will meet definitely when you will come in Bhopal.
Sure, sir. Thank you. Thank you very much.
Thank you so much, sir,
Thank
for
you.
the words, and stay tuned with us for with the same link. And I would like also welcome Dr. Harshita Tiwari for vote of thanks.
On behalf of DCRC Lab, I would like to express my sincere gratitude to Dr. Singour sir for his informative and interactive lecture. I believe that he might have instilled all the participants with the knowledge of computational drug discovery, and we will surely be benefited with the knowledge of computational drug discovery. So thank you very much, sir, for having us.
Thank you so much, ma'am.